In our last lesson about plants, we looked at the plant tissues. Now we're going to look at, remember that tissues that work together for a common function is called an organ. So we're going to look at plant organs and the three major organs of a plant: roots, stems, and leaves. This lesson is dealing with the roots, as you can tell by the pictures. Okay, so the types of organs in plant, know that they're the roots, the stems, and the leaves. Roots have a job of anchoring, of course, and it's either in soil or it could be onto another plant or it could be on an object such as a rock. So not all plants are anchored just in soil, although most of them is. And the root system's job is not only to anchor, but it's also to absorb water, because usually the plants cannot absorb water through their stems and leaves, even though they might be rained on. So it is the primary job of the roots to absorb the water that is needed for the plants. So look at the functions, be able to list these. Anchoring, absorbing, is absorbing the water. And the water then has to be transported, and that's by the sap-conducting tissues that we studied in detail about in our last lesson. And that is the xylem and the phloem. Now remember, which one is going to take the water with dissolved minerals? And that's what would be absorbed into the roots. Which of those cells, those vascular tissue cells, carries the water and dissolved minerals up the plant? That's xylem. And then in some plants, this is not all of them, there's going to be food storage. So roots are the organs of the root system. So you take all of the roots on one particular plant, that is known as a root system for that plant. There's two primary kinds of root systems. There are taproots and fibrous roots. And you can see here with the pictures next to each other the main difference. A taproot has one main root. Fibrous root does not. It has a whole bunch of them about equal in diameter. Now, does a taproot just have that one main root? No, they have a lot of secondary roots and so forth coming off of it, but they have one that's a lot, or it could actually be 1 to 3. It doesn't have to be just one. But a carrot is a good example that we like to use for the taproot, and grass is a good example of the fibrous root. Or if you're looking at the monocots and dicots, then you will look at the taproots and the fibrous roots. So commonly found in dicots are the taproots. Now remember, what is our dicot example that you're growing in your seed study? The lima bean. So that would fall under this. And they penetrate the soil with very little branching because there's one main branch. A lot of times these are the roots that will store food, because that's why they have that one main root, because that's where the food storage is going on, such as a carrot. Radish is a good example as well. So he has one or a few of the main roots that are thicker, they're longer than the other roots. And examples you need to know: carrots, dandelions. Fibrous root system is found mainly in monocots. Remember our typical example of a monocot? Corn. You have no main section. If you look at all the roots in the root system, you'll see that there's just a bunch of branching all over, and the roots are about equal in size in diameter. Not length, but in diameter. So there's a cluster of roots that are approximately equal in size. They branch several times. That's a key thing. You don't have a lot of branching when you have a taproot. Grass is an example, and of course our corn would be an example. Now let's get inside the root, and also on the external part. On the external part of a root, remember that a root is having to grow as it gets longer through soil. Now, what is soil made up of? Rock fragments. So can you imagine those new delicate tissues, those new cells being made to push down through the soil? If they came in direct contact as they pushed through the soil, they'd be shredded. So God put on them a root cap, a protective covering, so they don't get shredded by all the rock fragments as they move and push through the soil. It's a bunch of dead, thick-walled cells. That's what makes up the root cap. The root is covered in an epidermis, as is all those organs, the stem and the leaves as well. And the epidermis is always the outer covering, unless there's a cuticle, and there's no cuticles on roots. They're for protection. Remember, they keep things out that should not get in, also to prevent water loss. And the epidermis, because this is an important thing when you're looking under the microscope, it is only one cell layer in thickness. So the very first cell layer that you see in a root is going to be the epidermis. Now you might find some extensions off of an epidermal cell. And those long finger-like projections or extensions of the root's epidermis or epidermal cell, that is a root hair. And they are microscopic because the epidermis, to see the cellular structure, you would have to use a microscope. And one of the main things that they are doing is they're actually increasing the amount of soil that they can touch. They're increasing what we call surface area. And by reaching more surface area of the soil, they're going to be able to absorb the most amount of water with the dissolved minerals in it. Another thing is, remember, all the parts of a seeded vascular plant has vascular tissue. The roots, the stems, and the leaves. Now it's going to look a little differently in each of those parts, and that's what we're going to learn, how to recognize where's the xylem, where's the phloem, what do they look like in a root, a stem, and a leaf. So the xylem, remember, is the long, thick-walled cells, and it carries the dissolved minerals in the water up the plant. Phloem, those cell walls are slightly thinner. They're carrying the water and the dissolved food or glucose down to the roots, because the roots do not have the ability to make their own food by photosynthesis. They don't have chlorophyll. So the water conducting system in a root is found in the center. So it's smack dab in the center. That's where you would look when you're looking on a slide, smack dab in the center for the water conducting system, for the Fibrovascular Bundle, for the xylem and the phloem. Okay, so here's a good look at a root fibrovascular bundle in the center of the root. And there, remember, a fibrovascular definition is xylem and phloem surrounded by supporting tissues. Here's two examples of different kinds that we find. And you can see some of the parts that we talked about, the sieve tubes in the companion cells that are part of the phloem. So you got xylem and phloem. Now here's how you're going to tell the difference between a monocot root and a dicot root. So this is important, not for the quiz, but for lab. When you're looking at a dicot root, such as if you were looking at your lima bean and it's cut in a cross section, you're going to look for the fibrovascular bundle where? In the middle. And when you go to the middle, you're going to see the xylem is in the shape of an X. Whereas when you look in the middle for the fibrovascular bundle in a monocot root, which would be our corn, the xylem is going to be circular. There's actually in the very dead center, there's some pith, which is structural tissue there. And around the pith is where you find the xylem and the phloem. So it's going to be circular, not in an X. Now when you do lab, you're going to have to actually draw a pie drawing. And when you draw a pie drawing and there is an X in the middle, notice where the xylem is. The xylem is going to be at the very point of that pie. Then the phloem is going to be right behind it. And then you've got all the ground kind of tissue in between. And then you have the epidermis as the outer, and you might see epidermal cells originating out of, not epidermal cells, you might see root hairs coming out from an epidermal cell. Now, if that was the monocot root rather than the dicot root, remember that the xylem is in a circle. So the very front of the pie would not be xylem, it would actually be those pith cells. And then you'd see part of that circle, which would be the xylem, and then right behind it, the phloem. So that's going to be the difference in the roots, the pie-shaped drawing of the root. Here's again showing you where the root cap is, and where the meristem, that is the new growth. Meristems, we're going to talk about, that's the growth. And one place you find them is at the tip of roots. You also find them at the sides for lateral growth. And then dead center is where you have the xylem and the phloem in a fibrovascular bundle. And of course, if you look at this one, is that from a monocot root or a dicot root? It's from a dicot root because it has the X. Sometimes if you turn it another way, it looks like a plus sign. So it's either X or plus. Here, look in the very center. See in the reddish color, the X? That's a dead giveaway, dicot root, and that's the xylem. And then right beside, in each of those spaces of the X, you have your phloem. And then you have the supporting tissue all the way around it. So that's the dicot. That's going to help you in lab. Monocot, notice in the very dead center, you have not xylem and phloem. You have the pith cells. And then you see on a circle, you have the xylem, and then the next circle, you have the phloem in parts.